Hello, g'day, good evening. Thanks for listening. This is a medical cannabis show and I respect the law and I hope you do too. The information provided is for educational and scientific purposes. The facts are checked and the papers that have been discussed will be provided with links in the description below. And that's about it. So I hope you enjoy it as well. And today is an open topic, so nothing's been discussed prior to this stream. I'll be waiting for the comments in chat, and hopefully there'll be a cool question or a discussion topic so we can start, and then the show will be retitled. So if you're watching back, the title that you've got is because of what's happened in the future. And it's hard to predict what time that happens. So if you're re-watching it back, skip forward until you see some slides or the screen change. That's generally when it starts. And it's generally about 5 or 10 minutes. And if the stream's only gone for 10 minutes, that's because it didn't get a start today. That happens every now and then, which is all right. Because if that means if there's no questions or discussion topics, it means everybody's happy. So I'm happy too. And I'll wait till 2 minutes and then think about something to discuss. And actually, whilst that's coming up, the Paralympics is on, and I dead set support that so much. Those para-athletes, for what they've got to achieve just to try and, for instance, swimming, so they just don't get in the car or their bike and ride down the shop and get some food and stuff, then go to the complex. They got to probably get a lift there or go through wheelchair things or immobilized difficulties. It's so they're athletes just before they get to do their sport. So I take my hat off to those. So good on them. Good on them. And, all right, 2 minutes 20. I've got to think about something to discuss. There's nothing coming through in chat, so I have to really think. And that brings me back to the weather, because that's always changing. So that's all I can really think of. And we're coming up to the equinox soon. Another 4 weeks till everyone in the northern and southern hemispheres will be on 12 hours daylight, 12 hours nightlight. And then it quickly changes. It's, yeah, that's all I can think of to discuss. Far out. Wonder if I can end it earlier today. Well, the Paralympics is on. There's some support for it. Because I don't know what to talk about. I really don't. I've got a lot of things that I've already discussed, and it's hard for me to talk things twice. All my playlists are just thousands of lectures that I've already had. And it's some good information for you to go and look up if you're struggling with the topic. You can usually put it in the search engine and it'll come up with a few lectures for you to view. Oh, there's Genetic Memory Farms. Good evening, Genetic Memory Farms. Hope you're well, mate. I'm struggling for things to say, so if you've got anything that can start the stream off, you're welcome to add it. Otherwise, I don't mind having an early stream, just to keep things interesting. Yeah, because I don't know what to talk about, mate. What's a good topic besides the one that I really love and I'll talk about for ages, but what's something that keeps everybody happy? Because this is a stream for everybody, not for me. So that's why I have it's hard. Oh, here we go. Genetic Memory Farm says, I've got a topic if you want to chat about it. Practical THRIP options. THRIPS are horrible, horrible problems. Oh, geez. Yep, let's talk about that. So I've made a few videos on that. Or actually, I've definitely done one on how to kill them under the microscope. Did it a few different ways and timed it so you can see which products work differently. And the way to quickly get rid of them is cucumerous. That's a really good bio alternative to spraying and all that sort of stuff, which I don't support, because most sprays will kill all your beneficials as well. Well, hang on, genetic memory farms, we do some DE on the soil to diatomaceous earth and keep it dry? What can we do to make a hostile environment for thrips? Oh, well, let's look at where their larvae happens. Is it in the soil or is it on the actual underneath the leaves? So I'll pull up some slides. Let's go into thrips. Whereabouts, you reckon? Thrips. Trying to find where. Actually, it's probably in plants. It's in. I'll go back to plants, I think. Bio-pesticides for it. Yeah, I want to. Actually, I'll show that chart. That's a good start, I suppose. Here's a chart that shows some bios that you can source that will kill them. So over here on the left, they're the terrible long thrips, and you'll see they come to a point when you look under the microscope, or small. You can even see under the magnifying glass. So it'll tell you that it's them. And what this chart suggests is aureus, montgerensis, cucumerus, and hypoaspis. Hypoaspis is for the soil, montgerensis is for in the leaves area, and cucumerus is in the soil as well. And cucumerus was the winner. It was expensive, but that was the one. So montgerensis is really good IPM. So if you're coming into spring, you might want to get around a bit, because you can see it across the top here. Thrips, spider mites, broad mites, russet mites, white flies, aphids, scale, and then others that doesn't really happen much. Well, I suppose caterpillars and fungus gnats does happen actually quite a fair bit, fungus gnats. But the winner for the most of these two is montgerensis. You can see it's there, 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 there. And on this side is lacewings. So lacewing, 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 lacewing. So if you can source those two things coming into spring, if you know that you might have quite a lot of these bugs around, yeah, it's an asset. So I thought I'd show that just to start with. Western flower thrips, they're the bad ones. They're just hard to get rid of. It's because they reproduce fast. It's not very good. By the time you'll spray the larvae, or the pre-pupae, they've already killed this off, but they've already laid eggs again, and it's going through its cycle. So that's why they're really, really hard to get rid of. This is pretty cool. So I'll scribe in here. Of course, it's nothing. Just a bit of a fluke, eh? Was it? Thrips. Bio-pesticides for them. Let's see what this chart says. Bio-pesticides against various pests. So where would the thrips be? Disease, nematodes, mollusks, weeds, arthropods. Where is it? Miners, no. Okay, it's not on this chart, unless it's somewhere else. That's all right. There's a bit of a try. What else can I find here? Maybe let's go into the plants, because there'll be some in problems, viruses, bug problems. See if we've got anything under thrips. ST, tobacco, trichogramma. No. Oh. Aphids. And the flight bug problems, beneficial. Let's see what's in here. Oh, not much in there. There's on other things, but not under thrips. Garden beneficial insects. Let's see what this says. I haven't seen this for a while. Oh, there's your lacewings, your ladybirds for your spider mites, the snails, wasps, parasitic wasps, okay, and some beetles. Very good. In organic studies, there's some, so let's go through some of that stuff. Oh, I should see if there's any addition to any questions, actually. So I'll stop sharing and have a bit of that, and then we'll go back to some organic farming stuff. Oh, I didn't have to talk about diatomaceous earth. Oh, do we need some diatomaceous earth on the soil and keep it dry? Well, diatomaceous earth is like glass for microbes. So if they're penetrating into the soil a lot and you see them getting a lot of action in your substrate, it will help that reduce, because they won't be able to go through the layer, because that's how it feels on them. They're exoskeletons, the broken up diatoms. So what can we do to make a hostile environment for thrips? So that's what we're trying to come up with. So your IPM is what I'm just about to go through. Oh, there's Sunshine. G'day, Sunshine. There you go, mate. Oh, Genetic Memory Farm says, is part of their life cycle in the soil. It is for a grow student so they can start from scratch. Just need to ensure they are gone, don't come back. Hey, Southpaw. How you going, Southpaw? I'll see you, mate. And Raptor. G'day, Raptor. We're talking about thrips at the moment. Raptor. Yes, you can. I'm sure you can add some stuff. You just went through a battle. All right, let's look at the thrips life cycle. Where's my favourite wiki? Here we go. Now share screen. So it's part of the life cycle. So let's look at their life cycle. So we're talking about where it happens, where do they start, and what area to attack regarding these Western flower thrips. They're so blimmin' bad. Once you get them, you'll be so you'll know you got them, because they stay around for ages. Naming. Oh, look how long it is. Right, let's go down a bit. Morphology, evolution, tuning, taxonomy. Oh gosh, I've already passed it. Here we go. Thrips are believed to have, they live among leaf litter or on dead wood and are most important members of the ecosystem. Their diet often being supplemented with pollen. Wow, so you're into breeding, so that's not good. Number of thrips in the subfamilies. Most research, so I'll put this in the description later on to go through. Most research has found thrip species that feed on economically sufficient crops. Some species are predatory, but most of them feed on pollen. Wow. And the chloroplasts harvested from the outside of the plant cells. They prefer tender parts of the plant, such as the buds, flowers, and new leaves, besides feeding on plant tissue. The common bloom thrip feeds on pollen, Okay, very good. And eggs and whites. When the larva supplements in the diet, yes, there's a bit of a telltale sign. The curled leaves, you know something's going wrong, especially with the new growth. It can grow out of it, like you can get the new growth, suddenly it just goes good again, and then this section of the growth will stay like that unless you pick it off. So, and that's what you'd be hoping for. That's a good example. Damage to the plants. Thrips can cause disease during feeding. Yes, you'll see heaps of white dots on them. Heaps and heaps of white dots. Then you look under it and you'll see these little things moving around that look long. They're like a long looking, you squash them and all sorts, but they're their reproductive cycles so fast. Social behavior, no. Flight, no, they fly very good. Life cycle, here we go. Thrips lay extremely small eggs. So you can see there's 0.5 millimeter there, which is very, very small. There's one of the menaces. There's a little egg there. There's some larvae. So the life cycle's nearly been repeated in that little tiny photo. I'm going to try and script this. First, just skimming through this. The adult stage can be reached in around 8 to 15 days. Adults can live for 45 days. 45! So in that time, they're going to go and produce so much. Adults have wingless, for example, 90% of the population in spring with wings. Okay, so they get wing and wingless, very good. As pests. You can see from the photo, it's relatively large. Well, that one is actually not the western flower thrip. They look long and skinny like that. Some trick of grammar, I reckon. Biological insects such as fungi. You haven't got to that section yet. But yeah, that's how to we'll go through that. Bouviaria bastiana, or some metahizium, or some trichoderma even. Because trichogramma is a insect. It's a small predatory wasp. But what areas to target? Where's that? A lot of the areas happen in the, you can see their eggs are on the underneath of the leaves. So there's not much action going on in the soil. So to have diatomaceous earth that's sprinkled on the soil for your thrips, it won't do much. That's why getting hypoaspis, I'm still sharing, yep. Let's go back to that because that was very important. I'm going to pull it up again. It's down the bottom. It's down here, one of these ones, wasn't it? Also just been moved. There it is. Now I can't even remember the point I was trying to make. The hypoaspis is, that's right, that's a soil mite. So that concentrates beneath the ground, and above the ground is your Montgenesis and Cucumerus. And I found that this one, Cucumerus, just was rad. That was the one that really did the major damage. It was very expensive, and that's why I didn't get it at the start, because I'm not real rich, but all right. Oh, and where was I going to now? Oh, some other ways to tackle them. Hang on, was there a question? I can see on my tablet what's come through. Raptor says, having a wetting agent helped a lot with my IPM. Oh, good suggestion. Thanks, Raptor. Yep, helped to get it all through the cells and make sure it gets actually around the eggs and not just on top of them and evaporates. Good suggestion, mate. Genetic Memory Farm says, AussyCC, so ultimately I think I am going to throw out their soil and bring them some stuff of mine, new containers, and wipe down their tents. They have two. My soil has a population of hypoaspis. Oh, wow. Yes, well, that's really it's gonna help a lot. We just added. And do you think that will be sufficient, or are there other practical steps I should take? IPM spray. Oh yeah, yep. There's some more stuff you can definitely do. You're coming into winter, so you got temperature on your side. So the lowish temperatures doesn't help them. Are we still showing? No. Let's go back. The lowish temperatures doesn't help their reproductive cycle. So I'll go back and show you about that. Where's their temperatures they prefer? And through that, maybe I should just go through my organic farming, because there's some other stuff in there on them. And it shows about different IPM things you can do, like your pollen traps. It's not pollen traps. Your sticky traps. I'm trying to read at the same time and think. Sticky traps. Pheromone traps are really good if you got a lot of them. So that means if you can collect them, you can squash them all up, and that's going to create. So if you've got heaps of these, you can squash them all up, and then that'll make, well, a mix, and then that mix will go in the pheromone trap with a light above it, and then that attracts them because they've got hormones in it. And for instance, the males might be squashed up, releasing their pheromone, and attracts the females. So that's how a good way you can make your own one of those for free if you've got a lot of them, which I hope you don't. But if you've got an infestation, yeah, well, a good time is to leaf strip too, because see by all of their. That's a poor example of a leaf suffering. Fair dinkum. You'd see all the dots from under. I'm sure I got photos of it. Because where the eggs lay, I'm trying to get a photo of the eggs. Yep. Where the eggs are, there, they feed, and they have to feed off something. So on the opposite side of the leaf will start and kill itself. The hypersensitive reaction, where it starts to kill its cells, and then it expands out until it's killed the area where this is attached to. That's its defense. So that previous one, that's what you should see anyway, some dots above it with curling and stuff if it's bad. What was the question I was trying to get to? Just the different traps. Oh, and the temperature. Well, it is. The lower temperature is a lot better. So your northern hemisphere is coming into winter, so that's benefit for you to work with. Let's go into some other studies. Organic farming. No, there was some there. Organic farming practices. Where are you? This one. I did 2 courses. That's a basic one to this one, but still that has very good information in it too. Oh, there we go. Here we go. Just look at some of the lectures and see which ones are related to it. The IPM type thing. Introduction to Pest and Disease Management. Farming concepts. Oh yeah, from then onwards. It's lecture 17. So a bit into it. I just don't know what that is. Just for quick purposes, I'm just going to go here, and I'm going to try and script through. So we're looking at IPM. the signs of pest and disease, but we're focusing on THRIPS. So different ways to prevent and stop and manage THRIPS. This goes heavily into it, so I'm just going to stop on the bits that I think are suitable. Application for soil, cultivation, use of proper water, conservation for animals, selection planning, use of proper sanitary, cover crops, trap crops. Trap crops are very good. Marigolds are used to trap things, so they'll go to this plant instead of the others. How do you stop thrips? Once they're there, you've basically got them. So you've got to strip all the, here we go, here's a bit better. There's that light trap. There's your pheromone trap with a light on it and the sticky traps. So they're ways that you can stick in the room which will help, but it's not going to get rid of them one bit. So you take all, if you can afford to, take all the areas where the eggs are. So they're all around the leaves and stems. So I've manually gone and cleaned the bottom of them. So you can make up a wetting solution and, oh gosh, what's that solution you're going to say? It's a wetting agent with, so the wetting agent removes, like yucca, for instance, makes it like disinfectant. So you can use, actually you can use disinfectant too, very mild amount. Does a similar sort of thing, but it's got to be organic because there's some bad pesticides, like some organic ones, they use acids type thing instead of lime and stuff, but that's a way to clean them. So remove all the surfaces where they can potentially lay their eggs and where they are. So I'd be doing that. I'd have because they fly, you want a flying trap. So those yeah, I'll show you too. This is going to be a thorough one. So you want they're so amazingly effective. Come on. All right, 4-watt LED bug zapper. No, there they are, these ones. All these other ones are going to cost you top dollar, but if you get this ones, 20, 30 or 40 watt ones equivalent, but you get the LEDs, mate, they're incredibly effective, amazingly effective. It's a spin out, and with their low power consumption, it's nearly a must. If you don't have them, well, good luck. But anyway, they're rad, so I'll leave that there. Get back to is what we're up to. That's right. So there's some different traps. Here we go, some biocontrol. That's the way you're going to get rid of them if you've got an infestation. So by taking all the leaves off, bang, all the spots for where they can lay their eggs are potentially gone. Bio-augmentation is when you put microbes in. So you introduce trichoderma, Beauveria bassiana, or Beauveria species, Metahisium, trichoderma. So there's some ones that you could source, is those genus, and there's different species in those genus which work differently, but in general they worked well. Conservation. Okay. Population dynamics. Yes, if you want to get into it a bit more, you have to, when the numbers go low, doesn't mean they're gone. And that's a very sign where people just kick back and relax on. I think, you beauty, I'm not seeing nothing. Everything's growing good. No deformed leaves, no spots, no anything. But they're there. They're blooming there. They're just not showing you. So they're in a cunning area. The ones that have been killed off, they've learned maybe sitting next to new growth or just somewhere else. So be very, very careful when you get into the lower numbers. Oh, I missed out on Bacillus thuringiensis. That's for caterpillars, but still another IPM strategy. I actually use a little bit of that. It's not going to hurt. See, it does mosquito black fly control, so who knows if it might have adapted to different species that aren't in this list. There's different trichoderma, trichogramma, Hazarium. I use Hazarium, but there's a better one than that because that's the trichoderma species. Hazarium, there's a better one that you can get that's got hormones and it produces it as well as doing exactly the same thing as Hazarium. There's the Bouveria genus, and Bassiana is one of the ones they use. Actually, yeah, they use, it's a white muscarine disease. So if the insects go white, it's from Bouveria. And if they go green, it's from Metahisium, because Metahisium puts out the same muscadine disease. And biopesticides. So you can go and use these, like your neem, your pyrethrins, but they kill off beneficials as well. So that's why I tend not to use these. But if it was an infestation and you're looking at losing something very precious, I might consider using these. But yeah, I get upset when I've got to kill off my microbes because they're the things that work for me. And then it goes into it a bit. All right, that'll do. See what you reckon now. Half an hour I've been talking. I'm going to scroll up a bit. Do you think that would be sufficient? Or other practical steps to take on IPM? I didn't really go about the IPM. I'm sorry, I didn't talk about. I just talked about little things you can do. So remind me about that, please. Raptor says, what about hydrogen peroxide? Oh yeah, that's a good point. You can use hydrogen peroxide as a spray. If you're going to put it in the soil drench, you're just going to kill all the microbes because H2O2 is very bad against microbes. That's the risk you're running. So that is an option. I would not use that one. That's just nearly as good as, well, it's not isopropanol, but it does have an oxygen molecules in there which are beneficial. But because of the way it's structured, it's a poison type thing. It's not good to the microbes. Great question. G'day. Oh, Lene, hello, Lene. Nice to see you. Top of the morning to you, or top of the evening. GMF says, AussyCC sounds like if they persist into winter or come back, then I am buying some cucumbers after spraying down with some zymes, and I think have some powdered BT around. Oh, if numbers get out of control fast. I've had good experience with THRIPS, and over at least 12 months I've had them for in that one particular time, and I had to try everything, and every little thing that I've sort of said I ended up implementing because they were just so blimmin' hard. And then right at the very end, it turned out cucumbers just wiped them out, and it was $200. But that's why I didn't, or $160 for cucumbers. So that's why I didn't get it, because that's top dollar to me. And after I did get it, I saw numbers dropping. Everything was really good. And I went, oh gosh, if only I'd invested in that at the start, it would have saved me. Well, anyway, so don't leave it too late. That's for sure. And I hear what else I said. Raptor says, oh yes, I meant as a foliar. Oh yeah, no worries, mate. That's the H2O2 foliar. Yeah, well, it's going to dry stuff up. Before you're taking off leaves, why not try and kill the things on the leaves? Because the worst thing that could happen is you're going to remove the leaf anyway. So great suggestion, Raptor. So you could go and try that first, go around and spraying everything. Maybe put some towels across your substrate so it's going to be absorbed into it and not dropped into the substrate. to kill off the beneficials. And yeah, hit it hard. And also isopropanol. If you want to go down this route, isopropanol works well. But the trick is with that is you can't leave it on for longer than 2 minutes because it's going to kill the cells. It lyses them of their moisture. So they end up going hypertonic. Oh, gee, I hope I got that right. And the cells leach out, so they end up dying, they lyse. So that's the worst thing. So if you can spray it enough so the solutions go into and kill the eggs and your thrips that you want to kill and the larvae, and then spray it off with water so it doesn't be absorbed into the plant cells. That's the fine line that you're running with that, but that works well. So it's a good suggestion, Raptor. Thank you. Oh yeah, I don't know how the zymes, and the zymes are enzymes, and they're going to break down bits of material, and they're not going to break down unless it's got that chitinase in it, which is the cell walls of the insects. So that's the only enzyme I think would be relevant in this instance. Not to say that there aren't ones that do break down, like pyrethrin, the fatty salts of potassium acids. No, fatty acids of potassium salts also breaks down the cell walls of soft-shelled insects. So that's another thing that works too. So it's not to say that other enzymes don't, but I don't know if I'd be spending top dollar on that and looking and hoping for that to get rid of the thrips. Not to say it's not though, because who knows what's in it, unless you get a breakdown of every single enzyme that's in it. Raptor says, I like the towel suggestion. Yeah, to protect all the microbes beneath, just in case, and then thrash it, because why not? If you're going to be chopping all the leaves off, which is the way to get rid of them, or mostly, yeah, try it first. Try and spray it first, and you might just, and then you'll leave it, and then you'll see in 24 hours, like you put the towel down the substrate, and then you'll go and spray them with 70% ISO, and then wait no longer than 2 minutes from where you first put it, or you'll see 2 or 3 minutes. Because you might have tougher dermal tissue, where it might take longer to penetrate in. The waxy cuticles might have formed, so it might be a different situation. But yeah, and then you'll wait 24 hours and you'll start to see where the cells have died, that the hypersensitive reaction will take effect. And then you know which leaves to cut off. So that might be a better way to actually do it than just by cutting them off first. So, yes, Jesus, good to have good people in chat to help me out. Raptor says, I use a JADAM wetting agent, JADAM wetting agent, made with canola oil and potassium hydroxide. Wow. There you go. The oil's probably the main thing. Potassium hydroxide. It's going to give them some potassium. I wonder what hydroxide's going to do. Hydroxide. Potassium is a cation and hydroxide's an anion, so it's bound together and then it's held up in the oil. So I wonder if just the oil might be a good wetting agent. Anyway, that's a good suggestion again. And we'll get back to finding out other IPMs, things that from my studies that they've suggested. Oh, here's another top genetic memory farm says oil and drain cleaner, excuse me, had a super high pH base. Consider the pH with your sprays. Well done. Yes. Yeah, before spraying anything, you've got to always check that pH because what's those? There's some bad acidic ones. Yeah, you want to do. Oh no way! Look out! Got to look into that. Mace, g'day, mate. I was thinking about you the other day. Nice to see you. We're talking about thrips coming into the warm season. IPM strategies that you can do so you can prevent problems that are going to happen from different strategies that one does. And today's basically its topic on THRIPS. The canola oil is a brassica, which has an antifungal benefits. See all these double benefits? It's excellent. All right, we're going to go back to some of my organic farming studies and see what other IPM strategies they suggest. Neem oil, this goes into it and why it works. Neem, okay, application of neem. So it does a lot of things. Neem's really, they swear by neem. A lot of my studies are in India. So, and as a director, neem is it's the largest producing nation of it. So there's been lots of studies on it, and it works on lots of different areas. I'm sorry to those people in Canada, because it's actually illegal now. Pyrethrum, rotten, different things. With the sprays, you have to have a few different types because the bugs usually get resistant to one spray. What's this one? Oh, this is some homemade plant protections that you can make up yourself that they do with 5 these urine, neem leaves, and cow dung. And you ferment it and then filter it. And then that makes your spray. And then you test it, you pH it. And then you test for your ppms to make sure it's not over 1,000 or 1,500 because that'll burn the leaves. And then you can spray it, filter it, and then spray it. We're talking about IPM stuff. There's a soil drench that you can do similar way, just by fermenting it with cow dung because it's got all different IPM microbes in it, and the urine, which I don't think has many. Jaggery, it's a green manure, and pulse flower, they're sort of green crops that you can put in. Tillage plant spacings. These are ways that you can deter different microbes. Oh, back to the nutrient management. I love nutrient management. It's only, isn't it spin out? There's only 17 elements and 95% comes from the air. Look at this. Plant tissue contains 60 to 90% water and the rest is dry. And the dry matter, 90%, or actually 95, comes from the air. It's 45, 45. Anyway, not going into that. Come on, IPM. Nutrient compositions of some organic residues. No, that's more so into feeding now. Nutrient management, yeah, feeding. All right, let's try a different course. I stopped sharing. I'll send you. Oh, hang on. What else has been said here? I'll get back to another course. Raptor says, "Oh, yep, it's cheap enough." Sunshine says, "How important is a wetting agent in living soil?" Well, when it goes hydrophobic, that means water repels it. So the wetting agent, if you use the correct one, it will make it hygroscopic again, making the water penetrate. So instead of running across the surface, it'll go through it again. That's what a wetting agent does. And the same thing with your IPM is you put a wetting agent in your solution that you make up. And what that's going to do is that when you spray it on the leaves, if there's no solution, surely I've got this, I can explain this better with a diagram. When you spray it on the leaves, it's going to give a hollow effect of a length of, where's water? Come on. Fair, you can do all this different stuff. I just want the water molecule shape. When you spray it on, instead of going bulbous like normal, it'll elongate with the wetting agent, means it's going to reach more cells and a higher surface area. There's something here that's got it. What a shame. Out of all of these, water and life, maybe this one. It's hard to look at all the slides. photo I got which shows that, and that's the effectiveness of the wetting agent. And that's what it does. So I hope does that explain enough, Sunshine? So without going through all of these diagrams and getting, that's what it's made up of. Yeah, it's not there. All right, so let me know if that helped, or you want me to explain a bit more, Sunshine. Sunshine says probably a loaded question. No, it's no questions. It's all right. I can hopefully that's briefly explained enough, because there's 4 different types of wetting agents. You've got your positive, your negative, your plus and minus, and there's a neutral. So if you've got to get the right one to do the right thing. But in general, like yucca is a good example of a organic wetting agent you can use. Raptor says it can be used to quickly hydrate dried out hydrophobic substrates. Yes. And on the phytosphere too for leaves, and when you're putting in your sprays and IPMs and stuff. What's May say? Oh, there's a new medical strain out for Australian medical people. Amethysts. Very good. 25% odds of wedding cake and animal cookies. No way. You go 240 an odds. What's 8 to 10? Oh, that's your rating, 8 to 10. That's pretty blooming, and that's cost effective. Far out. There you go. There's some good medical information, good help. Good on you, Mace. Raptor says aloe vera has enough saponins to be used as a wetting agent for soil. Oh, nice one, Raptor. Aloe's very, very handy. Didn't you pulled all your aloes out ages ago, didn't you? And like, didn't you blended them all, or I remember you pulled it all out many years ago and blended it. I think you did. Sunshine says, if I were to use one, it would be aloe. Oh yeah, very good. Raptor says, I would best recommend that yucca or soap nuts. Ah, yes. Sorry, I forgot soap nuts as well. As a wetting agent. Excellent. All right, let's get back to 47. We've got 15 minutes to go, so I'll just go through another course and see what we can add for IPM regarding THRIPS. And put your questions and statements in now. Geez, the animal cookies, you got me going now. Organic farm. Let's go. I haven't been through this one in a while. Oh, is there any photos? Slides. Hadn't finished on the other one yet. Oh, because it went off track into quality. Oh. Okay. Oh, this has got some azos, really. Azos, brilliant. Anyway, come on, stay on topic. Here we go, some traps. Different right ways. Spike thrust method. Okay, just spike it into the ground. Tree banding. That's the way I didn't. Also, things climb up and down. And I've used, you know, when you clean out your implements, you've got the resin, the gooey, gummy resin that sticks to everything. I thought, why can't I put that around the base of the tree? Well, it works. So you can also get sticky things that they've done to ban them. But it's just another thing to stop things climbing up and down. And that's what it does. It traps it. It looks like that. It's pretty cool. Tree paste. Thing with tree paste is it can't breathe as good. Oh, sorry. This doesn't breathe as good either. But putting it on like a papery type of thing, and then you put it on top of that, like if it was, you wrap around, for instance, masking tape, and then put the sticky stuff on that. So it's not going to cut off those cells from breathing or make it mega thin. I don't know about that thick one. See that? The problem is that you might it might ring the tree, it's called, where all the cells underneath here are going to starve, and it might cause fungus and other problems. So it could actually be detrimental. Be careful with that technique. Neem again. Onions, some different sprays that you can go and do, capsicum, and okay. So, oh yeah, some different types of, what's this verticillium is an available in a wetting powder, is the wettable powder and liquid formation. This fungus is very effective on insects, such as the plant sucking ones. Oh, and that's what you want, because there's the cell suckers and there's the plant suckers. And yeah, there you go, the thrips. So verticillium lecanae is available in the wettable powder and liquid formations. It's a fungus. Okay. Something different. I've been through this in ages. It's good just to go through things again. Other practical for disease. Okay. Yeah, milk. Isn't that amazing? It's against anyway, stick to the topic. Copper. No, that's because it's for fungus. Sulfur. Fungus. Germination. It changed pretty quick. That's pretty cool. All the different. I like all the hormones to control it and different how. That's actually a very good chart. That different stages, what hormones to use. Germination process. No, dicot. So we can go forward a fair bit. It's got some good seed stuff. Bio-fertilizers. Wow, that's it. Very cool. There's a little land that you can use. And that's it for that. Just that. Okay. Fair income. Not many on that. Oh well. What other ones? Diseases of horticultural crops and their management. It's more diseases. I wonder if there's anything on thrips in there. I don't remember there to be. Plant pathology and soil health was a good old general one. Let's go through this one. Get out of here. Disease of horticulture and management. What have we got here? Oh, that's pretty cool. The content of, there's the paper on it, the content of calcium, magnesium, sodium, potassium of nuts and other foods. So you don't usually get, it's hard to get it's easy to get the NPKs of elements, but to get the CalMag, sulfur, silica, etc. is harder. So this is a pretty cool chart just to show that. So if you really want to get down to your nutrient management, this is very good. Look at cheese. 700 for calcium. Really? So can you use cheese as a calcium? That's per 100 grams. That's 700 milligrams per 100 grams. So that's nearly 1% of cheese. This chart says is calcium. 1%. That's a very large amount. And nothing else even comes close. Sardines is half that amount. Far out. Geez, I like information. All right, let's see what this reckons. Pythium, Septoria. Come on, IPM stuff. Trying to get into, not actual disease. Oh, that's because this is diseases. I'll skip a fair bit. It's going to show us all these different diseases in plants, and does focus on disease, not the pests. Bacterial. Let's see if I'm wrong here. Viral diseases. Bacterial diseases. Fungal diseases. That's it. It doesn't. So it was just all on plant disease. Oh, I should see if there's any. There's been a few questions in the chat, I think. So that's Raptor says, I have so many that taking 2 or 3 out doesn't put the dent in them. I have so many. That's good. It's good to have heaps of things. So if you do remove stuff, you got stacks. May says, look up Canada reviews. You can find the latest medical cannabis strains, prices for flower, oils, CBD gummies, wafers, hash, inhalers, and vape carts, and all other things that to help your medical needs. Sweet. That's good. Good help, mate. Help all the medical people. So I'm pleased to say that YouTube have finally recognised this channel as an educational and scientific channel, that the information is fact-checked properly, and everything, all the papers I go through are provided in descriptions. So to add other relevant medical information is highly vital. So good on you, mate. Keep up the good work. I've seen yucca is good. Yeah, you can grow it too. Even better. And you dry the root out, and that's what you use. It's got the wetting agent in that. I've got a chart of that. I have got a chart of that anyway. Oh, 55 minutes. Cool, nearly there. Let's see if there's any other studies that I've got has any relevance. So I've been through the diseases. Plant pathology. Soil fertilizers. That's got soil science. They have stuff about wetting agents. Trying to finish the IPM thing off. Oh, here we go. It's easily done. Viral diseases, bacterial, fungal, general life cycle of insects. Sweet. See if we've got anything relevant here to THRIPS. I'm not going to go into it heavily into that detail. Metamorphosis. That's of them, not of THRIPS. It's not looking good. Nymphs. Look at the size of the little baby things. Larva, next stage up. Okay, and the pupa. And then it's going to start and hatch. There you go. Come on, make this relevant. Oh, there might be one there, thrip. Battling something. Behavior of insects. What's the conclusion? Insects have a wide variety of fascinating life cycles and life history strategies, yet the basic forms that are used to build up these life cycles are relatively simple and easy to understand. Yeah, simple and easy. I don't like when they put that, because they're saying that it's simple and easy. Well, if it was, everybody wouldn't struggle. So they're basic techniques, but they're hard to adapt and implement at the right times. The way I would have worded it. Springtails, they're cool. See that little thing? They hop off that. Isn't that amazing? Columbola. Great, that didn't help. I tried. Plant pathology, what else is there? Pests of cereal crops. Hey, there's some. What? Here we go. I've got a slide on thrips. He's done it. He's done it finally at the end, at the flaming end. How do you know you got thrips? Oh, you can use a insect net and swoop your green leaf material and see what you find in the results. That's a Drosophila. Oh, I thought it was thrips, so I got to be excited there. And that's more of what you get. See that hypersensitive reaction, but it's to the dots. So you see heaps of dots, and you go, "Whoa, I better look underneath." Then you see heaps of action. Oh, look at those! Wow. You think, "Oh, wow, look how cool the trichomes are," but it's a bad disease. Eggs. So yeah, that's the eggs on the underside of the leaf is what you're trying to achieve from spraying with what we were talking earlier about those full-on ways of spraying with ISO and water. THRIPS management. What does this say? The population build-up of THRIPS is generally not large enough to cause. Okay, that's talking about a different pigeon pea. Insecticides such as that, or that, or that, used to reduce THRIPS. Yeah, well, I don't support those. I support the organic way. And we got the Western flower thrips mostly. Look at that. That's so scary. See how it comes to the point? It's a long, skinny looking. They're bad. Wow. In Eastern Africa, other thrips species are common. Okay. There's some ways that they suggest. They're dangerous ways of spraying. Look at all the big hazard signs on it. So it kills everything else. That's why I don't support those ways of spraying. Internode borer. It's like a top borer. Scale. That's it. Oh, that wasn't very helpful. I suppose it was helpful. It did say some stuff about thrips. Well, I don't think there's anything else in that course is going to have much about thrips then. What's this stuff? Reference links. Let's show you some reference links for thrips. Hopefully it's good. A couple of papers there. Not really that useful, to be honest. Glossary, script, summary. No. Could go through the script. Actually, let's have a look at the summary. Just to conclude a bit. Oh. Into the document. Okay. No. Out of there. Anything else that's interesting? Resistant ways. Yeah, it tries to treat them. Okay. So there is a fair few ways. If we go back into here. Biotechnology approaches of pest management. Integrated pest management. So yeah, I'll go through them. There's a few questions, I think. Questions or statements. Saponin. Yes. Let's go. Wetting agent. Howdy, howdy. Raptor says, can I put a good IPM? Oh, can I put a good IPM source? Can I put a good IPM source? Can you reword that, Raptor, please? Good IPM source. Yeah, integrated pest management. That's, isn't that amazing? That's just about what I was just going to go through. So coming out of, and for the Southern Hemisphere, is the IPM should be done now. You're coming into spring, and spring has sprung. What happened there? I want to share the screen. Didn't share the screen. Going back to this side. Oh, that's right, the IPM strategies. Analytical techniques involved in pesticide analysis. analysis and toxicological testing methods. I don't want to go that much into it. All right, let's have a look. Oh, there's not many. No, I'm not going to go into it because it's sprayed. So it's not the best. I support the organic approach. Biotechnology approaches in pest management. We'll show this one before I show the next IPM slide. Seed technology. Look at Bacillus thuringiensis under the microscope. Isn't that cool? And it does have an endospore. Really? Why does it say that you've got to reapply it every 3 months then? That's a good question. I'm not going through these old slides. It is generally accepted that the intensive use of Bacillus thuringiensis as generally engineered organisms will accelerate the selection pressure on insect populations to develop resistance. Propagation, technologies and IPM. There's different ways you can do your tissue culture for IPM. Because it's another thing, if you take a tissue culture and you put it grow it, and then once it's grown out of the callus and you've got your plantlets, you'll introduce problems and then see how you can fix it. So you don't have to put the big plant through the problems. There's a lot of tissue culture stuff. That's the shears. That's it goes through. Plant structure. That's it. Okay. This is just on tissue culture. It should be a reverted tissue culture. Right technology, I suppose that is tissue culture. Oh, what? 3. Continued use of insecticides from one chemical group can lead to the pest developing resistance and to these products no longer being effective. Very important. And that's, everybody has epigenetics and we've all want a way to try and survive. So it's pretty well known that if you do lots of sprays for things, especially like spider mites, you don't use one thing, you use 3 different types. With inoculative, this slide says, with inoculative release, a limited number of beneficial organisms are introduced at the start of the growing season. So when you're just exiting winter, or your last frosts are done, it's when Mother Nature comes alive and they all want to reproduce and it gets active. So that's the time that starts your IPM. The sterile insect technique is an area-wide IPM management program that introduces sterile male pests into the pest population to trick females into breeding encounters. There's another technique wrapped up. A green pest management IPM program uses pesticides derived from botanicals and other natural occurring plant materials. So they're the ways that I support, like your onion, capsaicin, and the other ones. Yeah, that's fascinating. That's the last thing. Yes, grasslands usually have higher soil organic matter than forests. Isn't that a spin out? And that's because of the nitrogen fixing bacteria fixing the lignin that's in the cell walls. It's a spin out, eh? Well, there are other fungicides controlled release, bionomic, stored grain, medical analysis figure, plant protection appliances, not even anything in there. Oh, yeah, I've got a few photos. Let's see what the photos are for this. So I usually keep relevant ones that I think are cool. Oh, it's just showing the hydrophobes next to the stomata, temperature. No. Try and keep it relevant. A few insecticides kill insects by interfering mechanically with their body functions. Okay. Yeah, that's one thing you've got to consider when you put your pesticides or your sprays on, which are hopefully organic, but they're still going to go somewhere. They're going to evaporate into the atmosphere, which can drop somewhere. They're going to run off and go somewhere, and they could seep, and they could go into areas where you don't want, or they could just be deposited and start killing all the bad things and letting all of the pathogens break out. Unmodified, modified. Conventional breeding, okay. Mutations, nope. This is pretty cool. Wow, that's very cool. This is more tissue culture stuff. I'm breeding. Crossing, okay. By continuing selfing, selections are made through F3, 5, 6 generations, and the best variety is selected. You select the best variety every time. Hang on. 143 different pesticides and 21 transformation products have been found in groundwater, including pesticides from every major chemical class in the lower 48. To continue, nearly every pesticide investigated has been detected in rain, air, fog, and snow across the lower 48 in the USA at different times in the year. So that's why I really support the organic approach for this reason. 98% of the groundwater is polluted in the lower 48, and there are no harmless substances. There are only harmless ways of using substances. Well, if you use glyphosate, that's harmful substance. So yeah, I think this was written for those people that still want to do bad things and just spray little amounts and think it's all right, it's going to be in the tissue, it'll break down, it won't transfer anywhere else. But there's a high amount of leaching, and as this slide shows, once it goes into the soil, it's absorbed elsewhere. Plant are bioaccumulators. They take it up into the leaves, into the vacuoles. It's dropped back down. In the vacuole still has that application of pesticide. Bang, it's leached into areas you don't want, killing potential microbes, beneficials. Or it's gone and volatilized up, vaporized into the atmosphere, falling back down as rain when it sticks to molecules. Or it could go and leach and just continue down with water into the groundwater, polluting all the groundwater, as what's happened in the lower 48. That'd be cool for GMF if you're still watching. Four more colours. Okay. TC. I think we're there at the end. I think that's it. Look at that cool way. It's a nice little setup, isn't it? The furrows and the channels. They still put their source, so they've got drip irrigation underneath it. That's pretty cool. Removable roof, possibly. Little tote-way. That's interesting. If oxygen should drop below 2.5%, if it dropped below 2.5%, conditions become lethal for insects, but fungi can survive with oxygen levels of 1% lower. So yeah, if you've got insect problems, righto, no worries. Just drop the oxygen to 2.5 and your thrips will be gone. How easy is that, hey? That is so flaming hard. But what's the oxygen absorbent? There was one of my exams where I had like moisture is, humidity is desiccant, Oxygen is anyway, you need too much of it to achieve that, so I'm not going to worry about that. What else have we got? That's it, I reckon. I could go through a few more, but I think I'd just be adding to stuff I've already said. Oh, we've got a few. Oh gosh. Howdy. Can I put good IPM? Oh, that's right, source link. Oh, I see what you're saying. I can't. How do I click on that? Oh, can you put this is what Raptor thinks is a good link for IPM. It's a shame I can't click on it, because I could put that in the description underneath. Anything in the genus Allium? I don't know of the genus Allium. Garlic would be best because availability and concentrate of allicin. Oh, okay. What's the best amendment for gibberellin? What's the best amendment for gibberellin? Gibberellin's a hormone, but you can get fugicarium or something like that. Gibberellium is the name of the fungus that produces gibberellic acid. So I'm not quite sure on what you mean for that. What's the best amendment for gibberellin? Can you reword that, Sunshine, please? Oh, Sunshine says gibberellic acid, oxen, etc. Oxen's different from gibberellic acid, though. What's the best amendment? Not sure on that, mate. I use microbes that produce gibberellin and oxen. They're individual microbes that produce different things. Elaborate on your question, please. Raptor says plenty in aloe, but I suspect willow is high as well. I love all these organic inputs. They're all so beneficial in different ways. Raptor says keep our salamanders, newts, and frogs friendly. Grow organically. Yes. Just listen to more your work. You really should do a scripted podcast on growing. Oh, thank you, mate. This goes to a podcast, actually. But it's scripted. You'd have to... it's got the... Now that YouTube's recognised this finally, it does put the subtitles underneath of my words. Thank you for the nice words, people. I appreciate that stuff. Raptor, did you say aloe has growth hormones? I see what you're saying. Hey, Heel Sauce, g'day, mate. Hope you're going well. Raptor says, I have many plant growth hormones at some time. So we're talking hormones. We were just talking on THRIPS in particular and IPM strategies and different ways to reduce THRIPS through organic practices, biopesticides, bioaugmentation, different ways to safely spray, etc. So that's really the guts of it. And now Sunshine has started to talk about hormones a little bit. But yeah, looking at different sources of hormones. Oh, here we go. Sunshine says, how do you boost growth hormones like gibberellins? Is what I'm asking. No worries, mate. You need something to produce it. So you have to have a source of microbes that produce that particular IAA oxen, and or a fungus can do it, or what's another way? Or it can be in the source of, like, tryptophan, for instance, is a precursor to oxen in the plant. So if you give it tryptophan and it wants to take it up, and it would help in that process. And if it was in the right process, signaling process pathway. But cut a long story short, microbes. That's how I do it, and it's amazing results if you use the right one. That's how gibberellic acid was by the Japanese discoverer, and his name was Fujirium or something like that, and he put it as that's the gibberellic acid producing fungus, and that's the name of it. Yeah, it's the microbes, mate. They're a winner. Once you get them doing their right thing, and if they can work at their conditions that they want, for instance, enrichment cultures. So if you've got one microbe, and how does it function? Well, it's got to function through just like a plant does and a human does. We all get our metabolism. So to make our metabolism happen, they require different things. So if you can give them the right 5 things that they require, you're on a winner. And that's what I'm getting into. Been experimenting like that and then for a few years. Yield Source says, what about ethylene and the effects it has on medical cannabis, specifically trichomes? Well, ethylene is seen in the plant as a stress hormone, so it's going to stress it out a bit more. So when it goes into senescence, it's releasing it a lot, and that's why it's senescing. And also at the start of your cycle, when the plant goes into flower, ethylene is released, and it's a way of keeping the plant female. It blocks when the female is produced, it puts out ethylene. When the male's produced, it's not. So if you burn putting a lot of ethylene out, you're blocking the pathways of the male, and it gives it femaleness, it's called. Yeah, there you go. Okay. But you ask the effects. Well, if you look at the signaling pathways of the trikes, salicylic acid and gibberellic and jasmonic acid are more of a hormone effector for trikes. So, he's added to it down the bottom here. So farmers mix ethylene and CO2 together in flour to increase yields? No. No. I would not think that would increase the CO2. Definitely does. But the ethylene, it's acting on the stress pathway. So the plant only releases ethylene when it's not that happy. I wouldn't think so. It's going to get a better secondary metabolite production. That's what it might do for that. And yes, it does induce it. It helps. Ethylene it's one of the 2 hormones used for seed germination. But I'd say the reason they mix it, the CO2 is for yields, and the ethylene would be for secondary metabolite production. So it's going to stimulate more stress in the plant, which is going to activate those pathways, which is to do with the terpenes and the trikes and stuff like that. That's how I think that works, mate. I could show some slides if it doesn't help. You got your caps lock on as well. Increases mineral absorption. I don't know about that one. This is reducing, you're talking about ethylene. Stimulates fruit ripening. Stimulates fruit. Well, it brings on senescence. Yes. Yes, and actually cut fruit supplies more ethylene. Once you cut it, more ethylene is released. What else? Like one bad apple ruins the bunch. Yes, because that's the way when it's cutting. Exactly right, mate. More ethylene is senescing and telling the plant to finish quick, finish. And like bananas. They'll pick the bananas early, and then they'll put them in a ethylene container, and they'll dose it up with I can't remember the ppm of ethylene, and it just ripens them mega fast because it's doing that. It's aging the cell. It's not very good. It prevents, what else does ethylene do? It prevents leaf expansion in germinating plants, but it can inhibit auxiliary bud growth and promotes senescence in leaves. Yeah, we'll talk about that. And chlorophyll degradation. Yep, it's not very good, but it's among the plant hormones, so used correctly, it can be very advantageous. Yield Source says there's a spray that increases ethylene production, and it's used for lateral branching. Oh, yeah. All these different things. And you look at the percent of the spray, and it's so low. Like the one for femaleness in plant, ethanol, no. I can't remember the name of it. It's something words along those lines, and it's 0.05% or something so low in it, but it's effective. So it's very beneficial. Actually, that's something interesting too about fruit ripening. Ethylene, it's also, I've got stacks of slides on ethylene. We've already been going for an hour and a half. There's another thing they did back in the day, how they discovered about how ethylene works with plant senescence, and that's with kerosene. Back at the turn of the 19th century, or the 1900s, they used to have the kerosene street lights, and they found that trees around it used to senesce faster, and that was because of the kerosene converts to a molecule that's very similar to ethylene and has a similar effect on the plant. It also says it inhibits stomata closing. Oh, okay. It inhibits stomata closing. All right. Yep. There's a lot of cool things that all the plant hormones do. That's a massive topic. I could probably talk just an hour on ethylene. I've got some. Remind me, Yield Source, come into another session. We're right at the end, mate. I hope that helped with that question, just briefly. But I'll show you here because hormones are everything, and a good botanist is supposed to be able to control any stage of the plant by using plant hormones. So actually, even just under here, plant science, I've got a massive, look at this. This is all the plant hormones that are relevant. The auxin, cytokines, gibberellins, abscisic acid, ethylene, salicylic acid, jasmonic acid, brassinosteroids, strigololactones, and florogen. I've got, and just under ethylene, we'll click under that. You have a look at all the slides. So it's all to do with ethylene, all these. And they're all extremely relevant. With a few papers in there too. Oh, you didn't mention the triple response. Seedlings. This is a triple response. Perfect example of it. So you get a shorter hypocotyl. It's thicker, and it has a J. It starts to hook, to give a J hook to it. That means that there's too much ethylene in the air at this process for germination. So it's called the ETR, ethylene triple response. That'll do. But yeah, in other words, I've got a lot of stuff on the different hormones because they're extremely relevant. So bring it up next time, mate. And I'll be very happy if we can make an episode on ethylene or, different effects on plant hormones on growth stages is also another good topic. Because I did an assignment on that, and it's just amazing how you can lengthen flowering, you can shorten it, like ethylene shorts it, cytokines lengthen it, different things like that. It's just really cool how you can control different growth stages from different hormones. And that's an hour and a half. That's been flaming long, and I haven't even talked about microbes. So thanks to everybody coming in. Put your stuff in right now because we're just about to leave. Put your stuff in chat. I appreciate all you cool folk for writing in chat. It's been excellent. Oh, we've got another thing. Yield Cess going to look into more. I believe it can make trichomes much longer. Maybe next time. Much longer. Okay, so you want to increase the stalked capitate cells. Stalked capitate cells. Interesting. Okay, yeah, because going from sessile to stalked capitate. All right, that's a fascinating topic. Genetic Memory Farms, great episode. Thanks, mate. Appreciate your nice words. Yes, I hope everybody rocks up this time next week in 167 hours time. And I'll be here doing the same thing to have good discussion. So happy breeding, happy growing, good health to you all. Hope to see you excellent people all again then. Thank you for listening. Happy breeding, happy growing, and good health to you all. Bye-bye.